Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about the neuron, and in particular about its resting membrane potential. I know that sounds a little tricky, but in essence, if you can see in this picture, this is a neuron. Here's the cell body, and here are the dendrites. What's really interesting about this, let me enlarge it so you can see it a little bit better. What's really interesting about this is that when a neuron is at rest, okay, it's negatively charged on the inside and it's positively charged on the outside. This is really important because when we talk about an action potential or a nerve impulse, we're going to be talking about a temporary switch of those electrical charges where the inside becomes positive and the outside becomes negative. And so it's important to understand that at rest, the cell has what we call a differential charge, a membrane potential. And this membrane potential has the ability to do work. Okay, so we can create a nerve impulse because of the fact that there's a separation of charge. It's sort of a chemical form of potential energy, a separation of charge. But what I want to say right out of the gate about resting membrane potential is that it couldn't be more, ironic that you would call it a resting membrane potential because the neuron works tremendously hard, expending a tremendous amount of energy in order to maintain that resting membrane potential of positive on the outside and negative on the inside. So I hope that's intriguing because we're about to talk about that right now. And so just a little review of a few concepts. This is the phospholipid bilayer of a neuron. And as you can see, sometimes there's proteins embedded in it, in the membrane, and they can translocate ions back from the inside to the outside. And they're very specific. There's ion channels for sodium, ion channels for potassium, and those ions can only go through those channels. And so they're selectively permeable, as you call them. And there's two different kinds. There's one kind that's open all the time, and it's called passive, and that allows the ions to move freely. And then there's active channels, which are gated. They only open when there's a chemical signal that interacts with them, a ligand, as it's called, and then it opens up. Or if the voltage changes dramatically, that will alter the shape of the protein and open it as well. So they're voltage gated channels and chemical gated channels, and both of those are active, meaning they can open and close. And so I want to make sure that this is really established. And I know maybe some of you have seen the previous video on ion channels, but I just want to make sure that when we talk about this resting membrane potential, we're all clear on the presence of where these ions are. So on the outside of a cell, okay, on the outside of a cell, there's a lot of sodium, a tremendous amount of sodium. Sure there is. Yeah, there's a lot of sodium on the outside of the cell. And on the inside, this is also on the outside, on the inside there's a lot of potassium, like this. It's a lot of potassium on the inside. Do you notice how it's low on the outside? So there is some out there, but there's a lot more on the inside. But overall, again, emphasis, going large here, emphasis. Overall, it's negative on the inside, positive on the outside. Now, why is this? Again, it has to do with the fact that there's a lot of anions on the inside in general, okay? Negatively charged organic molecules that are helping to create this. And also, the cell works very hard in order to generate this separation, and it uses something called the Sodium-Potassium Pump, and I'll get to it in a moment. But I just wanted to make sure that we're all clear that there's a lot of sodium on the outside, a lot of potassium on the inside. Now, with reference to these ion channels, remember how some of them, I'll just put some right here, some of these are open all the time. And as it turns out, sodium has just a few passive channels, which must mean that sodium has a tendency to leak into or diffuse into the cell. Okay, and that's true. But there are a tremendous amount of, if I can do this, there is a tremendous amount in the nerve cell, a tremendous amount of passive potassium. You see that? These are always open. So potassium has a tendency to leak out because it wants to go from a high concentration in the cell to the out. So it would leak out, leak out, leak out all the time, and that's a problem. Okay? Because you would lose this membrane potential if this were to occur. And so just to establish it, a lot of potassium on the inside, a lot of sodium on the outside. Overall, negative on the inside, positive on the outside. Okay, so a neuron at resting potential, again, ironic because the cell works hard to rest. So at resting potential, the concentration of potassium is greater in the cell, and outside it's greater in sodium. Okay, so again, here's the neuron like this. Here's the nucleus. A lot of potassium on the inside, a lot of potassium on the inside, a lot of sodium on the outside. A lot of sodium on the outside of the cell. Okay? But overall, it's negative on the inside, positive on the outside. Okay, so a neuron at resting potential contains many passive, as I was mentioning, potassium channels and not so many sodium channels. And so, as it turns out, potassium has a tendency to diffuse out of the cell because there's so much opportunity for it to do so. And so the anions, these negatively charged organic molecules, are trapped in the cell, and so it always maintains this sort of negative charge within. Now, the cells are very permeable to sodium as potassium, and they tend to leak out. And so, again, the reason being is, in terms of the membrane like this, if there is a potassium channel and it's passive, like this, passive meaning that it's always open, potassium, since it's high on the inside of the cell, has a tendency to leak out because it goes from an area of high concentration to an area of low concentration, tends to leak out. And so the cell has to work hard in order to keep the concentration of potassium high on the inside. Okay, that may seem mysterious, but I'll get to it ultimately. So this picture right here, I like it. It's a little busy though, so don't get freaked out. These little diamonds right here are potassium, and do you see how it's highly concentrated in the cell? These are the passive channels, so it's leaking out, leaking out, leaking out. There's a little bit of leaking of sodium in because it's high on the outside. So our conversation from this point on will be about a special kind of ion transport. Check this one out. It's called a co-transporter because, I don't know, have you ever been enter into a big tall building like a skyscraper, like downtown in a big city, where you have these glass doors where when one person's entering in, the door sort of rotates, and then the person exiting can also be in there at the same time, and the door sort of rotates, and some people leave and some people enter. This is what I'm about to talk to you about. There's an ion transport that's called a co-transporter. And you know how these are unique to potassium, these are unique to sodium? Check out this one. This one can allow sodium and potassium to move back and forth, but it's curious. It will only allow potassium to come in and only sodium to go out. And what's curious about it is the shape of it will allow three, let me just like illustrate that. It will allow 3 sodium ions, so here it's shown in red. It will allow 3 of these sodium ions to exit and 2 of the potassium ions to come in. So check that out. I mean, that's uneven. So there's 3 pluses coming out, which is kind of helping to generate that plus charge on the outside of the cell, and there's only 2 potassiums coming in, which helps to generate ultimately 2 and 3 would be a negative 2 over on the inside. Again, then the anions as well. Now, this particular co-transporter is called the sodium-potassium pump. And it's called a pump for a reason, because this transport, this ion channel, pushes sodium out of the cell against the concentration gradient. And it pushes the potassium back into the cell against the concentration gradient. That requires energy to go from low to high. And so it's going to involve adenosine triphosphate. It's going to require energy to do this. But the neuron is willing to use a tremendous amount of ATP in order to maintain this membrane potential. In other words, negative on the inside, positive on the outside. And it does it by using this sodium-potassium pump. It's sometimes called the sodium-potassium channel. So in general, here's the neuron, here's the cell body dendrites, here's the axon, here are the collaterals over here. And so take a look. On the inside it's negative, on the outside it's positive. Do you see that? Here's the concentration of these ions in question. So as it turns out, potassium has a tendency to leak. And so potassium would be leaking out. And so what would happen is the membrane potential, the negative on the inside, the positive, would eventually run down if this were allowed to occur. And there is some sodium diffusion in as well, because it wants to diffuse in. But the sodium-potassium pump grabs 3 sodiums and pushes them out, and then grabs 2 potassium and pulls them in. It requires energy to do that in the form of ATP. So again, like that door going into a hotel, 3 sodiums would be exiting, and 2 would be rotating. Potassiums would be rotating in. It's kind of cool. And so the sodium-potassium pump's able to do this, and it expends a lot of energy. And let me see if I can help to illustrate it. So you got the phospholipid bilayer like this, like that. There's some phospholipids, 3 of them. And what's interesting is you can go really huge right here. 1, 2, 3. Now remember, the phospholipid bilayer is impermeable to ions. And so that's kind of neat, because then the ions just can't go free willy across the cell. So they need to go through a protein. So as it turns out, what the cell is doing is it's allowing, because of its shape, it's allowing sodium ions. Let's go red here. It's allowing those sodium ions to attach like this. And as they attach, the positive charge of the sodium is altering the shape and allowing them to move across the cell membrane. You see that? It's allowing them to move. And it's like, well, how is that allowing it to move? Well, it's also the result of ATP. Now, ATP has 3 phosphates, and so when it loses one of its phosphates like this, when a negative charge, it allows the molecules to be translocated across the cell membrane. As a result, it becomes adenosine diphosphate. So the neuron really needs to generate a lot of ATP. So it has a lot of mitochondria, the neuron does. So therefore, a nerve cell requires a tremendous amount of glucose in order to produce a tremendous amount of ATP. So the brain requires a tremendous amount of blood sugar. Why? Because the sodium-potassium pump requires a tremendous amount of energy. When those sodiums reach the outside, it then will allow, over here, it'll allow 2 potassiums, shown here, 2 potassiums, to then come back into the cell, like that. So 2 potassiums come into the cell, 3 sodiums exit the cell. And as a result, It's more positive on the outside and more negative on the inside, relatively speaking, proportionally. Okay, and so here's a better picture than the one I drew, perhaps. This shows the place where the three sodiums would get in. Here's at ATP, so the phosphate attaches here. The sodiums leave, and the potassiums shown in purple would then enter in. So it's pretty neat. And so let me actually take a moment while we're talking about the sodium-potassium pump. Let me see if I can call up a couple of images here. I happen to have some right here that you could see. And so this is a pretty cool little animation right here showing, like, okay, here's the nerve impulse coming down the dendrites. It's being picked up right here. And what you're going to see ultimately is a close-up of the neuronal membrane. And the reason that we need to see a close-up is because these sodium-potassium pumps are found in the neuronal membrane. And it's like, well, where are they? I mentioned in a previous video that these pumps are located in specific areas. The sodium-potassium pumps are not found so much on the dendrite, but they're found along the axon hillock and axon in general. So check this out. So we're getting closer and closer and closer. And now you can start to see what we're talking about, which is the resting membrane potential. There's a lot of potassium on the inside, a lot of sodium on the outside. It's positive out here on the outside, it's negative on the inside. But you have this sodium-potassium pump. This yellow right here is ATP, so check it out. So the potassium comes in, and then 3 sodiums go out. And it's and then when this comes in, it knocks off this. So then these potassiums, the sodiums come in, and then they allow it to leave this way. So it's pretty cool. It's a co-transporter, and the cell uses a tremendous amount of energy in order to do that. And again, the reason it wants to do that is to maintain the negative on the inside and positive on the outside. That's really important. And so this is also a little animation showing this as well. So inside the neuron, the sodium concentration is low, and it's really high on the outside. So sodium's high on the outside. But if it's high on the outside, the cell would have to really use a lot of energy to push it out. So the sodium-potassium exchange pump, it's sometimes called, because this carrier protein's a co-transporter. So when the sodium docks to this active site in the ion channel, what happens is the phosphate group of ATP then attaches. It changes the shape, which will translocate the sodium ions out. So it requires energy because it's against the concentration gradient. And then when those exit, it opens up an active site over here for the potassium ions. And so 2 potassium ions then attach to the protein. Since they're positively charged, that will influence the shape. And so it'll kick off the phosphate, and then as a result of no phosphate, it sort of resumes to its normal conformation, which was like this. And so the potassium then come in. So the point of this is to make sure that all your potassium is not leaking out. So that's rather important. And so I wanted to make sure that we're on that. Okay, let's see if I can get back to our slideshow on this. And so resting potential, I think you can see now what I was getting at earlier, takes a lot of work to rest. And so the resting potential is all about generating an electrical potential, a potential to create a nerve impulse. I haven't explained the action potential yet. That's coming in a separate video called Action Potential. But ultimately, it's this separation of charge that creates a potential between the inside and the outside of the cell. So again, when the cell is resting, it's pretty permeable to potassium. So the potassium kind of leaks out. Little sodium comes in. So overall, the inside of the cell, this is a fairly important number. The inside of the cell is negative, as you know. Let's make it negative. Whoops. Make it negative like this. So the inside of the cells are negative. It's positive on the outside. But what is the charge on the inside? It's negative 70 millivolts. And so that's going to be something that is important when we talk about action potential and how that is generated. So it's pretty negative inside the cell, and the cell works hard to keep it that way because, again, it's negative 70 on the inside. So if you had a voltmeter and you were able to check it, it would be negative 70 on the inside. It works hard because, as it turns out, potassium's kind of leaking out, so the cell has to work hard to pull the potassium in and push the sodium back out. And so the sodium-potassium pump, as it's called, helps to keep that membrane potential just where we want it, which is negative 70 on the inside relative to the outside. And that's because it's pushing 3 positives out and taking 2 positives in, as you can see here. This is a great diagram. It shows the sodium-potassium pump, but it's kind of weird. But it shows it as it's cycling around. I kind of prefer an animation personally, but this is pretty informative as well, showing that the sodium is being pushed to the outside against the concentration gradient, and then the potassium is being pulled in against the concentration gradient, thus maintaining the resting membrane potential. So, hope you enjoyed this video on membrane potential in the neuron. Thanks for watching.